Artificial sweeteners have long been promoted as a healthier alternative to sugar, offering the promise of sweetness without calories, weight gain, or blood sugar spikes. They are widely used in beverages, desserts, snacks, and tabletop sweeteners, often marketed toward individuals seeking weight management, diabetes prevention, or general metabolic health. However, emerging research indicates that some artificial sweeteners may influence glucose regulation and gut microbiota in unexpected ways, raising questions about their long-term metabolic impact. While these findings warrant careful consideration, they also offer opportunities to better understand the complex interplay between diet, gut bacteria, and metabolic health, and to make informed choices that maximize the benefits of reduced sugar consumption. One of the most intriguing insights from recent studies is the impact of artificial sweeteners on the gut microbiome. The gut contains trillions of microorganisms, many of which play critical roles in digestion, nutrient absorption, and metabolic regulation. Evidence from both human and animal studies shows that certain non-nutritive sweeteners, such as saccharin, sucralose, and aspartame, can alter the composition and activity of gut bacteria. These changes may influence glucose tolerance by affecting microbial metabolites, gut barrier function, and signaling pathways that regulate insulin sensitivity. In animal models, mice fed high levels of artificial sweeteners developed marked shifts in microbial diversity, including an increase in bacteria associated with impaired glucose handling, and these changes were accompanied by measurable increases in blood glucose levels. Remarkably, when the altered microbiota from sweetener-fed mice were transplanted into germ-free animals, the recipients also exhibited glucose intolerance, highlighting the causal role of gut bacteria in mediating these metabolic effects. Human studies, though less controlled, provide corroborating evidence that artificial sweeteners can influence glucose regulation in ways that vary between individuals. Some participants consuming high doses of sweeteners experienced reduced glucose tolerance and altered postprandial insulin responses, while others showed minimal effects. This variability appears to be linked to baseline gut microbiome composition, suggesting that the metabolic consequences of sweeteners are highly individualized. From a positive perspective, this finding illustrates the remarkable adaptability of the gut microbiome and the potential for dietary interventions, such as prebiotic fibers or diverse whole food-based diets, to mitigate any negative effects. By nurturing beneficial bacteria through nutrient-rich foods, individuals may be able to maintain stable glucose regulation while still reducing overall sugar intake. The mechanisms behind artificial sweeteners' effects on glucose metabolism are complex. In addition to altering microbial composition, sweeteners may influence gut hormone signaling, particularly incretins like GLP-1, which help regulate insulin release and appetite. Changes in microbial metabolites can modulate gut inflammation and intestinal permeability, which in turn affect how efficiently glucose is absorbed and processed. While these pathways may appear concerning, they also underscore the body's intricate ability to respond to environmental inputs and highlight opportunities for positive dietary strategies. For example, pairing low-calorie sweeteners with fiber-rich foods, fermented products, and balanced meals may support healthy microbial function and reduce potential impacts on glucose handling, demonstrating that sweeteners do not exist in isolation but interact dynamically with the broader diet. Animal research has provided additional surprising insights. Some studies have shown that even relatively modest doses of artificial sweeteners can provoke shifts in gut microbial communities that persist over time, and these changes can influence energy metabolism and fat storage. Yet, the same research also reveals that dietary context matters enormously. When sweeteners are consumed within diets rich in whole grains, fruits, vegetables, and prebiotic fibers, negative metabolic effects are less pronounced or even absent. This suggests that artificial sweeteners are not inherently harmful, but that their impact is modulated by the surrounding nutrient environment, emphasizing the importance of a holistic approach to nutrition rather than focusing solely on single additives. From a broader perspective, artificial sweeteners continue to provide important benefits for many individuals. They allow for reduction of added sugar intake, which is strongly associated with obesity, dental decay, and cardiometabolic disease. When used judiciously and in combination with fiber-rich, minimally processed foods, sweeteners can support weight management, improved energy balance, and compliance with dietary goals without necessarily provoking negative metabolic outcomes. In addition, understanding their effects on gut microbiota provides an opportunity for proactive dietary management, Incorporating diverse plant-based foods, fermented foods, and whole grains can help maintain microbial diversity and metabolic health while enjoying the convenience and sweetness of low-calorie alternatives. Overall, the emerging evidence paints a nuanced picture. Artificial sweeteners can influence glucose tolerance by modulating gut bacteria and related metabolic pathways, but these effects are highly context-dependent and reversible. The gut microbiome's adaptability offers a pathway to mitigate potential risks, and integrating sweeteners into balanced, fiber-rich diets can preserve their benefits while minimizing negative consequences. Rather than discouraging their use outright, this research encourages thoughtful, personalized dietary choices. With careful meal planning, attention to overall nutrient quality, and awareness of microbial health, artificial sweeteners can serve as a practical tool for reducing sugar consumption, supporting weight management, and promoting long-term metabolic resilience. In this light, these sweeteners are not a metabolic hazard, but an opportunity to better understand the dynamic interactions between diet, gut microbiota, and glucose regulation, ultimately empowering individuals to make informed decisions that support both taste and health.